This video is on sexual reproduction and is the first topic in looking at maintaining life. Makes sense because sexual reproduction is how life begins for many of us. In this particular video, we'll look through how sexual reproduction occurs through meiosis and fertilization, and look at beginning to think about some of the processes as to why it evolved by looking at the genetic variation that entails. So to begin with, sexual reproduction can be summarized like this. We're using here an example of humans, but this would be the same for plants and many other species as well. So in general, what we're talking about here is a cycle in which we go between a life cycle. It includes haploid cells, gametes here, and diploid cells, which grow multicellular organisms. So for humans, almost all of our life cycle occurs in this phase here, where we are a diploid zygote cell initially that grows through mitosis and development. So the zygote is a single cell. Growth occurs through cell division of the mitotic type to produce an adult multicellular organism. Then, in some very specialized cells in the sexual reproductive organs, the ovary in the female and the testes in the male, meiosis occurs producing haploid gamete cells. So gamete cells are just the sex cells, also single-celled organisms, single-celled, well, they're not quite free-living organisms, but they're not exactly attached either. So we have these single cells, the ovum or egg, and the sperm cell here. And these are called haploid, meaning this end, their haploid number is 23, so they contain 23 chromosomes, which is of course half the amount of a diploid cell, which has 46 chromosomes. So here we have 23 pairs, making 46. Here we have 23 individual chromosomes. There are no homologous pairs in the gamete cells. To restore diploid number, fertilization occurs, which of course is the fusion of those two gamete cells to produce a new zygote, and the cycle can continue. And so this is what we call sexual reproduction. Rather than just producing a clone, which would be one diploid cell from an individual producing a new organism, which can occur in plants, for example, clonal propagation, which is a form of asexual reproduction. Sexual reproduction requires gametes from two separate organisms to come together to make a new organism. And of course, that new organism is genetically different to both of its parents because it shares genetic information. But there are also a number of other forms of variability that occur, and we'll look at those. So to produce a gamete cell, this is how it occurs, and this type of cell division is called meiosis or meiotic cell division. Very similar in some ways to mitosis, but some really clear key differences. So the first thing you might notice is that the titles for the phases are the same: prophase, metaphase, anaphase, and telophase. But in meiosis, these occur twice to produce 4 daughter cells. The other key difference happens in metaphase, and I'll get to that in a minute. So in prophase 1, very similar, the nuclear membrane dissolves and the chromosomes condense and align, but they align quite differently in meiosis to mitosis. There's a separation, formation of 2 new cells, and cytokinesis. Then the chromosomes align again, the chromatids separate, producing 4 cells this time. In fact, this second round of division is very much like mitosis, but it's really this step here that's quite different and leads to the genetic variability that we see. In fact, already at this stage you might have noticed that there's a little bit of red in the blue one, a little bit of blue in the red, and that we've got red on that side and blue here, and red there and blue here. So these are some forms of variability. So we can see that these 2 cells, if we're just looking at the colors of their chromosomes, are quite different, and all 4 here are different to each other. So how does that occur? Well, the key thing happens at this first metaphase stage. So if we're looking at mitosis, for example, the homologous chromosomes align side by side with sister chromatids on either side of that cleavage plane. Apologies for the poor resolution on this image, but it's the best I could find. So in this case, once they divide, each cell will get 1 copy of that homologous chromosome, and these are identical copies to each other, sister chromatids, so they'll end up with the same on either side. For meiosis though, the homologous chromosomes don't align side by side but together in a structure called a tetrad. And so in this case, the white one's going to go in this direction and the black one's going to go down here. So initially these cells are going to be quite different already. Secondly, you see that there's a little bit of switching over, which we call a recombination happening, crossing over of those chromosomes, and so they're actually switching parts. And so not only is this one mostly white, but it's got a little bit of black, and this one's mostly black with a little bit of white. Again, these daughter cells already have genetically different information to each other, but also to the parent. And this is a key source of variability. So let's go through those sources of variability in detail. So the first source of variability in sexual reproduction is mutation. In fact, this is a source of variability that occurs in all types of cell division, including binary fission and mitosis. Whereas if you have a mutation that occurs in an early embryo or in a body cell in a fully formed organism, that might affect some cells or one or a few cells and could lead to cancer, for example, but has no effect on the gametes of that individual and is not passed on or inherited. But if you have mutations in what we call the germline, in the production of those germ cells or gametes, whether it's the sperm or the egg, that will affect all of the cells that come after fertilization, and that new organism, every cell in its body will carry that mutation. Furthermore, half of the gametes it produces might have that mutation as well. So it could not only be passed on to one generation, but subsequent generations as well. And so even though mutations occur in all types of cell division, it's particularly relevant when we have mutations happening during meiosis, because they will have consequences for all subsequent generations. Independent assortment is one I referred to when we're looking at which side the white and black chromosomes. Here we're just using blue and red. But you can see on this side the paternal in blue and the maternal align all on the same side for this particular cell. And then when that divides, this cell gets all the blue, this cell gets all the red. It could equally occur that we have a mix of red and blue, and therefore you end up with a mix of red and blue in the daughter cells. And this is for only 2 chromosomes. Imagine when if you had 23 pairs, how many different combinations of reds and blues switching around you could have. And then once these divide, you can see that there's variation in those daughter cells. So that's called independent assortment, and that mixes up the homologous chromosomes from both parents into the gamete cells. The next form is this recombination of chromosomes called crossing over. So when we align in what's called the tetrad here, tetra is related to number 4, so we have 4 chromatids together. Remember, these are homologous chromosomes, red and blue, and each has replicated to make sister chromatids. So because these are homologous, they have the same genes in the same locations but not necessarily the same sequence. But because they're homologous, they could have large sections of DNA that are identical. And because of this, base pairing can occur between strands, leading to breaks and joins that lead to recombination or new combinations of these. So what might happen is if there were genes here in the blue part of the chromosome, different alleles to what was happening in the pink, those could actually be transferred over to here. So this blue and pink chromatid, although it was originally genetically identical to the pink one, is now genetically different. And so we have 4 genetically distinct chromatids now, all pink, pink with a little blue, blue with a little pink, and all blue. So by crossing over, we've made 4 distinct different chromatids of this chromosome. Let's say it's chromosome 7, now 4 different versions of it. Finally, when fertilization occurs, those chromatids that might have recombined or independently assorted to make an egg cell or a sperm cell now meet up with another gamete. Now that combination of these 23 single chromosomes meeting up with 23 single chromosomes to make 23 paired chromosomes will lead to a combination that is unique and probably never occurred in human history. And so by looking at mutations, independent assortment, crossing over, and then combining those through fertilization, we end up with a genetically unique individual with 46 chromosomes in 23 pairs, but this combination may never have occurred before. And so this is one of the very powerful things of sexual reproduction, is that it has just the right amount of variability to create offspring that resemble their parents but are also unique and genetically diverse.